Hello, Anin. My name is Dr. Christine Lalonde. Welcome to our poster entitled Adult Phenotypes Associated with Prenatal Exposure to Radiation. We have no conflicts to declare, and this study was supported by NSERC, MITAX, and the Nuclear Innovations Institute. Ionizing radiation has sufficient energy to remove an electron from an atom or molecule, producing a highly reactive ion or free radical. Direct damage to DNA can occur by the release of electrons, but indirect oxidative stress may also occur by the production of reactive oxygen species and promote the methylation or hypermethylation of genes. During pregnancy, exposure to ionizing radiation usually occurs for diagnosis or therapy and are deemed necessary or safe. Exposures at high levels, such as greater than 1 gray, during development can be deleterious. However, at low doses, the changes in offspring are not always apparent until adulthood when facing a stressful environment. This study exposed BALB/C mice who were pregnant to one of 4 conditions, seen on the top right panel. Sham, 0.05, 0.3, or 1 gray of ionizing radiation on gestational day 15, which is also the late trimester. 2 male and 2 female pups were randomly chosen from each litter to make up a sample size of 8 males and 8 females per condition. Once the pups reached adulthood at 17 weeks, they underwent behavioral testing, the pour salt swim task, where they were placed in a bucket of warm water to swim for 5 minutes without an escape platform. Here they were measured for immobility or float time. The open field, where they were placed in a novel box and allowed to explore for 5 minutes, looking for exploratory behavior. And the social anxiety task, where animals are exposed to a stranger mouse behind a safety wall and are measured for attempts at interaction. 1 day post-testing, animals were sacrificed and neural tissue was dissected and prepared for genetic analysis. In Figure 1A, at the bottom left panel, the results of the pour salt swim task show the total time spent immobile for floating in seconds. The 1 gray, or 1000 milligray, offspring spent far more time swimming compared to all other groups, and the 300 milligray, or 0.3 gray condition were far less active. In Figure B, for the social anxiety task, the measure here is the average number of approaches to a stranger mouse. Both the 0.05 gray and 1 gray conditions were more active than the sham or 0.3 gray offspring. In Figure C, for the social anxiety task, this behavioral measure is the rearing activity, where a mouse stands on its hind legs to explore. Both the 0.3 and 1 gray animals were far more active in this figure. Figure D is an illustration of these 2 tasks. In Figure E, male brains are represented on the left and females are on the right. In the 0.05 gray condition, there are 9 downregulated genes in the cerebral cortex and 10 in the hippocampi. These genes are related to ROS regulation, glucocorticoid receptors, cortisol regulation, DNA damage and tumor suppression, methylation and synaptic activity, as well as microglia and apoptosis. Aside from 1 upregulated gene in the hippocampi, the 0.3 gray condition is similar to the 0.05, with a robust downregulation of genes. In the 1 gray condition, however, we have no response in the hippocampi and a different responding genes in the cerebral cortex, predominantly in synaptic activity, ROS, and cortisol regulation. In conclusion, we see a significant disruption of coping mechanisms and disinhibition, as well as dysregulation of important genetic pathways. These changes are non-linear, however, with differential responses in the 1 gray group, indicating a hormetic response that could be seen with other types of prenatal exposures to different potential toxins, where low doses tend to be adaptive in nature. Thank you for listening to this poster. Miigwech.